So it was really an interesting paper that one of my students, Alex Schnell, who did this, and she was looking at when a mouse is undergoing through autoimmunity, what kind of Th17 cells are present normally at homeostasis and during an autoimmune reaction. And she did single cell RNA-seq of Th17 cells from different organs, including lymph nodes and spleen and gut-associated lymphoid tissue, mesenteric lymph nodes, and brain in case of autoimmune disease of the brain. And then analyze their T cell receptor sequences as well to see how similar or related they are. So first thing that she noticed is that the Th17 cells, depending on which tissue they come from, they assume the transcriptome of the tissue they come from. So that is the most overwhelming feature in the cell, that somehow tissue imprints an unique transcriptome and they cluster together if they come from that tissue. That's number one. Number two, when she was doing clonal analysis of T cell receptor of the Th17 cells, and she found that obviously during the autoimmune reaction, there is a major class of Th17 cells that are very pathogenic, and they produce IL-17 with other pro-inflammatory cytokines like interferon gamma and GM-CSF. And these cells go to the target tissue. In this case, they go to brain and induce autoimmunity. But at the same time, there is a very small stream of Th17 cells that are induced, and those cells go to gut. And these are non-pathogenic stem-like homeostatic Th17 cells that you see in the gut. And these gut-associated Th17 cells have their own job, but they are the providing reservoirs for the future attacks, for the relapses. And they, if you activate them right way with IL-23, these cells will assume a pathogenic phenotype, effector phenotype, and they will land up in the brain and induce disease. So this provides you actually a very nice, interesting paradigm and explains that every time you have an autoimmune reaction or an infection, you have effector T cells that are generated that clear the infection, but you also have a small stream of the stem-like homeostatic T cells that go and invade the gut, and they become gut resident. And these must be providing you then the supply of T cells for future. We know that these cells do exist in humans. We know that they exist in the gut of the humans because we have taken the transcriptome of what we find into the homeostatic Th17 cells. We see them in the gut-associated lymphoid tissue that have been single cell seeked. And but we do not know in humans whether they are the reservoirs and contributors to relapses and remissions. What I would love to see is how to convert, what are the factors that will convert the pathogenic Th17 cells and into the homeostatic ones. So that in an autoimmune disease reaction, all you got to do is to block those factors, and then instead of inducing autoimmunity, they will actually go into the gut and promote barrier functions and all the good things, but not induce disease.